When you think of proteins, you probably think about diet and building muscle. While that is a part of what proteins do, that's not all they do. Proteins are responsible for carrying out your body's cellular processes. Enzymes, specialized proteins that help your body perform the chemical reactions to keep you alive, assist in breaking down food, building new cells, and even aiding your body in fighting off infection. As enzymes are essential for everyday biological processes, it's important to understand how they are made. Hi, I'm Anthony, a member of the DU Quark. We are a student-led journal that helps make science accessible to viewers like yourself. Today's video focuses on a foundational concept in biology known as the Central Dogma. Thank you for watching. Your body stores the instructions to make proteins in your DNA. Different genes within your DNA code for different proteins, but the DNA does not make proteins itself. First, double-stranded DNA is turned into an intermediate molecule called RNA, which has the same nucleotide building blocks as DNA, but only possesses a single strand. The RNA meets a part of the cell called the ribosome, which reads the RNA and uses its code to assemble the target protein from different building blocks called amino acids. This system of DNA to RNA to protein is called the Central Dogma. The first step of the Central Dogma, going from DNA to RNA, is called transcription. DNA's code is made up of 4 different letters which bind to each other. A binds to T, G binds to C, and vice versa. A special enzyme reads a single strand of DNA and transcribes the sequence into a complementary strand of RNA. At the end of transcription, you are left with a single strand of RNA copied from the original DNA. This RNA transcript contains the sequence that codes for the final protein. The second step of the Central Dogma converts the RNA synthesized during transcription into a protein. This is called translation. A special enzyme complex known as the ribosome binds to the RNA transcript and reads it 3 letters at a time. These groups of 3 letters tells the ribosome what amino acid, or building block, to add to its protein chain. Each 3-letter combination codes for a specific amino acid. This strand of amino acids is the final protein product, which, after some light polishing, is ready to do its job. In addition to the standard progression of DNA to RNA to protein, there are exceptions to the Central Dogma. Some viruses, like HIV, have a special enzyme that makes RNA from DNA. This exception makes these viruses highly infectious, as they can convert their RNA into DNA and insert it into the host's DNA. This can result in lifelong and recurring infections. Overall, the Central Dogma is one of the foundational principles used in molecular biology and has given us many tools to understand human health. In fact, the Central Dogma is the basis for studying genetic diseases. This classification of disease is explained by detrimental mutations in your DNA that change the final protein product, making it less effective. One example of this is muscular dystrophy, a group of genetic diseases that cause progressive muscle weakness and degeneration of muscles. This genetic disease can result from a single base change in the DNA which alter the protein sequence, leading to lifelong complications. From the DNA in your cells to proteins that keep you alive, the Central Dogma is the story of how life builds itself, letter by letter, amino acid by amino acid. The more we learn about how our cells read and carry out their genetic instructions, the better equipped we are to intervene when things go wrong. So the next time you hear about a new gene therapy, a breakthrough cancer treatment, or a vaccine, remember, it all starts with the Central Dogma.